So we are studying the TAM receptors in the specialized tissue macrophages of the brain called microglia. We got into this originally because one of the TAM receptors that we study, a protein called Axl, is very strongly upregulated in microglia during the course of Alzheimer's disease. So we studied this in both human AD, but mainly in mouse models of AD, of amyloidogenic mouse models, where we looked at the effect of removing Axl and Mer from microglia during the course of disease in those amyloidogenic mouse models. And the main conclusion that we found were, well, there were several. The principal conclusions that we found were that without TAM receptors, microglia have great difficulty even detecting plaques in the brain. So they behave almost like normal microglia would in a healthy brain. They can't see the plaques very well. They don't react to the plaques very well in terms of their morphology. And most importantly, they don't do any phagocytosis of the plaque material. So normal microglia, when they see plaques, they spend a lot of time eating up, internalizing plaque material. The microglia that don't have the TAM receptors, Axl and Mer, the two ones that they express, don't do this. So we anticipated, since they're not eating A-beta material in the brain, and before we had started our work, most people assumed that microglial phagocytosis of plaques, which other people had seen, was actually constraining the growth of dense core plaques. Since this wasn't happening when we knocked out Axl and Mer, we assumed that we would have many, many more plaques in the brains of these mice that didn't have Axl and Mer. And in fact, when we looked, we actually had fewer plaques. So the phagocytosis is reduced about 10-fold, but instead of having 10 times more plaques, we actually had half as many. And we followed up on that observation. And the principal conclusion then from this, from our work and from work in some other laboratories, is that the dense core plaques of Alzheimer's disease, which are stained with dyes like Congo red and thioflavin S and methoxyXO4, these are one of the two defining histopathological features of AD, that these dense core plaques don't form spontaneously. So the A-beta doesn't have the ability to form these plaques. They're actually formed by microglia eating up looser forms of the plaques, less aggregated forms of the plaques, aggregating them and condensing them in the lysosomes of the cells, and then spitting them back out to make the dense core plaques. So the fundamental conclusion is that the dense core plaques of Alzheimer's disease are actually constructed by microglia, and they're constructed by microglial phagocytosis.